to understand and kind of see things with their traits. So some of that we're going to look at as Mr. Burns was talking about. And as he said, first of all, we kind of need to understand what it is we're doing. All right, so up here, this is something that we've seen really similar. What are you seeing in this image? Quickly tell your table. What is this? Go. Let's try it again. I didn't hear much. Okay, tell everybody at your group, what is this image? Go. All right, group 3. What do you see in this image? Chromosomes. Group 5. What's true about these chromosomes? Perfect, thank you. So it's their chromosomes. Now, as she pointed out, we don't know which one of these is from the mother and which one is from the father. Just looking at it. But we know that we have to have one from each parent. This is a normal cell. This isn't in a sperm cell or an egg cell. It's in a normal cell. So I'm going to show some quick symbols. Anybody know what this symbol means? What's it mean? Actually, this one is the female symbol. So, and then this one is male. Again, like I said, we don't know by looking at it. So here's what I'm going to do. Just like what Maddie was saying, we know, and I'm just gonna guess. And so this is one pair of chromosomes. This is all chromosome 1, and again, there's one from the mother and one from the father. That's true here. That's true here. I'm not going to draw 22 more of those, but you get the idea that if I have chromosomes and they're paired up, that's what we call diploid. Just kind of as a review. One from each parent. And as Mr. Burns talked about when he showed that image with shoes, pair is always 2. Di is always 2. Think about dioxide and all the things from chemistry or other things that you might know. All right. Down below, you're going to see some other terms. And 2 of them, or a couple of them, relate to this image. I'm going to zoom out just a bit because I still want to look at this image. So the first term, and this is what one of the things you're going to have to do. Sorry, let me scoot it down. On the assessment, that project that you'll start next week on Monday, is you'll need to identify some of these terms in your patient that you're looking at. It won't be Maggie, but it will be a patient like Maggie who has some sort of disorder. You won't know what the disorder is. You're going to kind of have to figure it out, and then you're going to have to give some explanation. One of the things is you're going to get a karyotype. Again, this is a karyotype that shows the chromosomes. It's just an image that shows the chromosomes. All right. So there are autosomal chromosomes. These are the chromosomes that are numbers 1 through 22. These are the ones that just determine genes that are everywhere in your body, but not about your biological sex, right? It's just about what is in your body. So it's any chromosome that's not a sex chromosome. So in this case, for humans, that's chromosomes 1 through 22. So all of these are autosomal chromosomes. All 22 pairs of those are autosomal, just in your body anywhere. All right? Sex chromosomes are just the other, it's the 23rd pair. Now, here's one thing that gets confusing when we draw images of chromosomes and we talk about chromosomes and when they're in pairs, is they're going to be either X or Y chromosomes. Now, people think that an X chromosome is in the shape of an X and a Y chromosome is in the shape of a Y. It is not, right? So an X chromosome is just longer. So that's a pair of X chromosomes. Because that's going to be one of the things that might be confusing when you look at the karyotype for your patient. Oops, that's an I. All right. Now I'm going to zoom back in a little bit. If I look at a pair of Y chromosomes, or a pair of Ys, an X and a Y, if you have a pair of Y chromosomes, it happens, but they don't survive. And so for this one, this is what an X and a Y chromosome would look like. Neither one looks like an X and neither one looks like a Y. The Y chromosome is just shorter. If I have an X and a Y, that's going to make a male. If I have two X's, it's going to make it female. Again, the X chromosomes, like this one, that one is going to be a female. Because that's Maggie's. Yep. None of this other stuff tells me if it's male or female. Zero. Nothing in there says male or female. So here's what I always think is confusing. Draw this line in here. It is not when we talk about, oops, thanks. It's not when we talk about these chromosomes that are connected at a centromere. This is one of those reasons that it always irks me when people just make them as an X, because actually it's got this shape to it, right? Where the two things come together. It can look like an X, but there's that centromere that's joining them together. Okay, so that's what I just want to make sure if you're looking at an image, which sometimes you'll see, sometimes we've seen images of chromosomes that are homologous, that they're paired, that will look like an X.
Really, they're just like this. You take these and they kind of bend together.
Kind of like the images we looked at in our baggies yesterday.
It looks like an X, it's not an X. It's
right.
Just these two kind of squeezing together.
Yeah. Okay. So I just want to make sure that we understand that. All right, so that homologous pair is just two chromosomes from the same size and shape. So this is, these are in homologous pairs. I know I'm doing lots of arrows. This is also a homologous pair. All it is that we just have pairs of them. And so we'll just do a quick little image down here. And so you're just going to draw them, same sizes. All it is. It's all, like the word sounds really confusing, and we're going to get some words that look really, really similar. So don't confuse homologous with another word that we're going to see in just a bit. You guys are doing a great job of putting down a lot of this first hour. So thank you. Okay, allele is the thing that we talked about a lot yesterday. All right. So for example, the things that we talked about is that it's the different version of the same gene. This is what we show with the lowercase. Or capital. So in the ones where we did this with these images here, we looked at, there was an E, and that stood for that earlobe. And whether it was free or attached, right? So like you might have the hanging earlobe or the one that's attached. So you either have this kind of an earlobe. It's great art.
You gotta draw the rest of the head.
I know.
Otherwise, it's a weird C.
And that's going to look like this on your head. And that we showed with the capital E or the lowercase e. And attached was the lowercase e. And we talked about the hair texture. Straight or curly. And so we used a capital H and a lowercase h. In just a second, we'll talk about why one is capital and why one is lowercase and which one I didn't just randomly assign them. So we'll talk about that. All right. So the next one is dominant. What that means is that it appears even if there is one dominant allele for that. So what that means is if there is an H capital and an H lowercase, or there is a capital H and a capital H. So that there's the allele for the curly hair and the straight hair, or both curly hair. That means it's going to show curly hair. So it doesn't matter if there's 1 or 2. It's dominant. If it's there at all, it's taking over, and that's what it's going to be. If it's 2 lowercase h's, then no.
So I'm showing it on the chromosomes, because remember, this is a physical location where these alleles are.
Oh, thank you.
These are genes on a chromosome, different copies. Each of you has some mixture of these.
All right, so recessive is kind of the opposite. It only appears if there are 2 recessive alleles. So this one was the capital H. This one's the lowercase h. And it only happens if there's the HH, lowercase both. And that gives us the straight hair, as Mr. Burns just showed us on the board. What that means is even if there's a lowercase h in there, if there's a capital H, it's not going to show. It's going to be curly. Now, we talked about yesterday that there's sort of this gradient between curly-er and straight-er, kind of in that middle. And so we're not going to get into that just yet. I'll give you a moment to write this down. Yep, when you got it, yeah, go ahead and turn the page just so I can see you're all ready. I promise in a little bit it's going to get more interesting. But I want to make sure that you've got this so you can refer back to it, especially when you're looking at that project next week. Mr. Burns and I were like, okay, what's the stuff that we need to make sure people have easily accessible so that they can do the project? All right. Sounds like most people have turned the page. Okay. Codominance is one of those things that we are going to get into. And codominance appears when you've got 2 things happening at the same time. So let's say I'm just going to do a flower because it's easier for drawing purposes. So if I look at a flower petal, like the blossom, let's say I have one that's white. And then I have another one that's going to be a red color. Or this is green, so let's just say green. I don't know very many green petaled flowers there are. And what happens then, okay, good, is that when I cross those 2, what happens is I'm not going to get a white one and I'm not going to get a green one. I'm going to get something in the middle. And so what that's going to give me, oops, sorry. Just kidding. Ignore this one. So if you started to draw that one, that's the next one. What you're going to actually see are maybe it's stripes or maybe it's polka dots or different things, right? So there's going to be maybe just a line in a white petal, and it's going to show them both at the same time. So this one's going to be white and green.
So here's the same thing shown on the chromosomes. We write the alleles a little differently. We put the color above, but the alleles are the same because they're both impacting the trait the same way. If you get two versions of the white, you're going to have white flowers. If you get two versions of the green, you're going to have green flowers. If you get a green and a white, then you have striped flowers. This is what it would look like in the chromosomes. You still have two copies of each gene.
It's not dominant. Neither one is dominant. Neither one takes over or is like, I don't know, more dominant than the other one.
So we don't use capital and lowercase for this.
And I'm just going to put down what Mr. Burns has up there so that if I've got a color, and as he said, we show the alleles differently. So if I have a white and a white, that's going to give me a white flower. And there are different terms that we use for writing out the color or the trait versus the letters, and we'll talk about that in a little bit. But if I had the C and the G plus the C and the G, that's going to give me a green flower. And if I have the C with the W plus the C with the G, it's going to give me green and white. All right. Now it's the thing I started to do. So again, if I have a white flower and a green flower, oh, sorry.
You should do green and red because it's clear what color is partway between. Oh, it's too late.
Green and red.
Oh, between red and red.
Yeah. Sorry. Yeah. Yeah. I thought about that. That's usually the So now that's where I'm going to have the flower that's going to be a light green. Or if it was red and white, like Mr. Burns was saying, it's going to give me a pink flower. Do we have a red?
Oh, that's fine.
I mean, I was just going to show another example, but I like that you've got that up here.
Otherwise,
So
it stays the same as the one that we did that was codominant. The only way you can tell these apart is by looking at a whole bunch of these flowers to figure out if these traits are codominant or incomplete dominant. You got to look at them. Like, do they come out pink or do they come out red and white spotted? Do they come out green or white or light green or green and white?
And there are some flowers where either one can happen.
So we're just looking at different ways we can inherit our traits. We can inherit them so that we get either one or the other of our parents' traits. We're just like one or the other of our parents. Or They can be inherited where we have some blending of the traits of our parents. And this is true for all living things, plants, animals, humans. Well, we're animals.
And that's what happened here. So this was our CW and our CG. Our CW and CG. Now, you can kind of tell we try to pick letters that kind of make sense. It's like, oh, the C stands for color, and then the G stands for green. Or the C stands for color, and the W stands for white. Sort of like what we did with the hair texture. I did a capital H for hair and a lowercase h for hair, but the uppercase, the capital H, was for the dominant one. The lowercase is for the recessive. If I kind of mix those up, and if I did like an S for straight and a C for curly, it's hard to see which one's recessive and dominant. So we use those when it's straight up recessive or dominant. With codominant and incomplete dominant, it's a little different. Don't worry, if this is confusing, we're going to practice it, okay? It's probably confusing, and that's okay. All right. So sex-linked happens on the sex chromosomes. So it is something that happens on the X chromosome. If you're a female, you have 2 X chromosomes. So that's an X and an X because this is a female. If you're a male, it's going to be the X and the Y. Now, this is an interesting one. This is kind of the one that, for me, selfishly, is interesting because things like color blindness, which I'm colorblind, is that I only need color blindness on this X chromosome because I don't have another X chromosome. If it's there, it's going to express it. My mom is also colorblind, but that meant she had to have it on both of her X chromosomes in order for it to appear. Her mom, my grandmother, had it only on one of hers, so she wasn't colorblind. So it's kind of one of those things that's like all of my brothers have to be colorblind. If I had any sisters, they wouldn't be because my dad isn't. So it's just, we'll talk about where do you get your X's and Y's from, and that'll maybe make a little more sense. So X-linked, if it's recessive, females need on both. If it's a male, you only have one X, so it's got to be just on that one X. There are some things that are X dominant, but they're super rare. And the ones that I can think of are incomplete dominance, like hemophilia. All right. I'm going to get into some language stuff that maybe will help you. So hetero means different. If I think about heterosexual, it means a male is attracted to a female, right? So those are going to be different. When I think then about doing alleles, that means if I have a capital F and a lowercase f, they're different because I've got one capital and one lowercase. So they're different. So one is going to be dominant and one is recessive. So this is my dominant. I showed it with a capital letter. And this is recessive. So if I have a dominant and a recessive together, am I going to see the dominant trait or the recessive trait? The dominant, right? Because like we said, with dominant traits, I only need one of the alleles for it to show that dominant trait. All right. Homozygous dominant. Homo means the same. So that means they're going to have both the same, either two capitals or two lowercase. This one is also going to be the same, either two capital or two lowercase. If it's homozygous dominant, that means it's going to have the two dominant alleles because they're both the same. Homozygous recessive is going to be both those recessive alleles, the lowercase allele. So in this case, I'm going to have an F and an F. And I'll talk about why we're using an F for this. And so both of these are dominant. So that means the dominant trait is going to be shown because that's all there is. For homozygous recessive, that's the only way we can see the recessive trait because the dominant one would take over. It would dominate. So for this one, it's going to be the lowercase f and the lowercase f. Now again, we chose the letter F. At first we were going to use the letter C, and it's like, well, you can't really see the difference, no pun intended, between a capital C and a lowercase c. So we used lowercase f. And I'll tell you why we were deciding between C or F. So both of these are recessive. And that means that's the only way you see a recessive trait. Oops. Only way you can see that recessive trait if there's two recessives. All right, so we're going to come back to Maggie. Why do you suppose we chose an F for Maggie? Do you remember what Maggie has? You remember the disorder she has? So Maggie has cystic fibrosis. And like I said, we could either have chosen a C for cystic fibrosis, but we said that's not a good choice, or the F. So we chose that. All right. Now, in the case of Maggie, she is affected. She shows the disorder. So in the case of Maggie, Maggie has cystic fibrosis. And again, that's why we chose that F. So in the case of my family, I am affected because I have color blindness. My mom is affected because she has color blindness. My brothers are affected because they have color blindness. So they have the disorder. I hate thinking of me as having a disorder, but it's true. It's not the normal. And then with unaffected, her parents don't have cystic fibrosis. Mm-hmm. Oh, thanks. But they might carry it. So my grandmother, my mom's mom, is unaffected. She's not colorblind, but she carries it. My dad is unaffected, but he doesn't carry it. Incidentally, my dad was the only one in my entire family growing up who wasn't colorblind. He always said he felt like the same person in the crazy house because we all saw the same colors or lack thereof, and my dad was like,
Tan van.
the tan van, the green wall, the gray wall. So yeah, so the carrier is the one who has that recessive allele. So in the case of Maggie, the cystic fibrosis is recessive. Sorry, guys. I know. That means both of Maggie's parents carry cystic fibrosis, even though they're unaffected. Because it's recessive, she has to have both alleles in order to have cystic fibrosis. So they're unaffected. So like I said, my grandmother is a carrier. So a non-carrier would only have the normal dominant gene. My dad is not a carrier and he's unaffected. Remember, that's where we use that little f. This part is where people are going to get mixed up on the assessment, or at least have previously. The difference between the words genotype and phenotype. Genotype just talks about the genes. And those are the letters. So for example, this is a genotype. This is a genotype. This is a genotype. I'm just looking at the alleles. I'm just looking at the letters. I'm not saying anything about the trait or the disorder. Like, I'm not saying if this has cystic fibrosis, which one does. I'm not saying that. I'm just talking about the letters. The phenotype is that physical trait. That's how I always remember or teach people to remember is phenotype starts with a PH and physical starts with a PH. It's that physical, what's in the body, that's the trait. We're not using those letters of capital and lowercase. We're just saying the name of the trait. So this one's in words, not in letters. Doesn't say what the letters are. All I'm saying, do they have that trait or not? Are they colorblind or not? Not getting into my mom's got X colorblind, X colorblind. I've got X colorblind, Y. That's my genotype. Two more, friends. We'll get there. And we'll take a break after this. So dihybrid cross is when we're looking at two traits at the same time. When we looked at these examples, we talked about two different chromosomes for two different traits. We talked about the E chromosome, the E allele, the E gene, and we talked about the one that's got T. When we're doing a dihybrid cross, we're just talking about two different traits at the same time. For example, if I talked about hair texture and free earlobes. Now, I don't know why we would want to talk about somebody who has curly hair and free earlobes. Or we talk about straight hair and free earlobes. Or we talk about curly and attached. Or straight and attached. Oops, attached. All of them talk about 2 different traits, and they're just 4 different combinations. Before we talked about 2 different combinations, straight or curly, free or attached. And now we're talking about the combination of the 2. So we're just looking at, this case, earlobes and hair texture. So pedigree is just the family history. So for Maggie, what is her family's history? So what do we know about the parents? What do we know about grandparents, aunts, uncles, cousins? So we're looking at the history of cystic fibrosis in Maggie's family. And sometimes we don't know about their family history, either because they're adopted or we just don't have the information about them. So when I was telling you about my family with color blindness, that was my pedigree. I would just map it out. And if you want to look and see what a pedigree is, just flip the paper over and you'll see some examples of what does it look like. We're not going to do it yet. We might do it on Friday. We might not. I think it's interesting. Okay, take a break. You got this down, take a break. If you're missing any of them, come on up and write down any that you're missing. If you're gonna take a all right, so bless you. Grab me a new packet, actually. Oops, perfect, I have 3. All right, so for this one, now we're gonna actually remember how yesterday we drew the squares, we divided into 4 parts. This is where we're gonna actually use some of them, but now we've got an actual situation, actual scenarios. So for this one, we're using these, they're called Punnett squares. That's just what they're called. I actually don't know why we call them Punnett squares. There's probably a person named Punnett who took Mendel's ideas and put them into a square. And this is the way of figuring out probability. It's all about predictions and likelihood. That is the purpose. It's going to show both the genotypes, and then we have to decide the phenotypes. The square itself just shows the genotypes, but then we also have to say what is the phenotype because of that genotype. Again, like I said, they sound really similar and they get confused easily. All right, so in this one, we've got Zea mays. That's just corn. That's just what we call corn, like corn on the cob. There is corn that has, most of it that we see in the store has yellow kernels. All of them are yellow. There is corn that is purple, and actually there's lots of different colors you can get on sweet corn, on Zea mays. And purple is actually dominant, which is kind of weird. You're like, well, why don't we see the yellow? Why don't we see more purple than yellow? That's a selective breeding question.
People know how to use these Punnett squares to make the kind of corn that they want.
And we'll get into that in try 3. Okay, so now we've got one that has yellow, and we've got one that's got purple. We're going to cross the two. Now here's the thing. Like I said, we choose letters. There isn't a specific letter for it. So what would be a good letter that you think either we should or shouldn't use on this? Any letters that it's like capital and lowercase look too similar? Usually we want to use a letter that's somehow connected to the words in the situation. Like we use the C and the W to show color in white, and the C and the G to show color in green. So oftentimes, we could use the C, but a capital C and a lowercase c look too similar. Well, because we call it ZMAs, or this could also be color, we could, because it's ZMAs, use a capital and a lowercase z. They look too similar. So I don't know. Let's use M for maize. Also, maize is a word that we often use to talk about corn in general. Those look different enough that I'm going to be able to see it better, especially when we're handwriting. We just choose letters. We choose letters that are going to make sense. Because if I use a letter like P or H, well, what does this have to do with H? Where in there is something that's going to remind me that that's an H? All right, so which one did we say is going to be dominant? Purple or yellow?
Yellow. Oh, no, purple.
Okay, so if it's purple, here's what I want you to do. I want you to write the letter below purple, capital or lowercase, and write capital or lowercase next to yellow. Just do it on your paper right now. I'm going to write one letter for the purple kernels and one letter for the yellow kernels. Which one's it going to be? All right, here's what we're going to do. Capital, you're going to point up. Lowercase, you're going to point down. And we're going to use this a lot today. So for purple, are we going to use a capital M or a lowercase m? Point with your finger. Yep. See a lot of capitals. Why? It's dominant. So we're going to use a capital M. And the yellow kernel, are we going to use capital or lowercase? Show it, point with your finger. All right, yep. So I see lots of lower cases. So we're going to use the lowercase m to show yellow kernels. Again, we just chose the M because it's the maize. We didn't choose P or Y. We could have. But we try not to choose one that's going to show the other one. That's going to be a word of the trait. All right, so we're going to talk about for this one, it's going to be homozygous. So what I want you to do is look back at your vocab that we just did. All right, write down the genotype. Just do it right now. If it's homozygous, look at your vocab or just remember it. What is the genotype? What are the letters, capital or lowercase, that we're going to use? All right, now you have to use 2 fingers. For purple, if it's homozygous, am I going to do a capital and a capital and a lowercase and a lowercase? Show me with your fingers. What am I going to do for those? Give me 2 capitals, 2 lowercase, a capital and a lowercase. All right, we said it's dominant. Purple is dominant, so we're using that capital M, and it's homozygous. It means they're the same. So for purple, they're the same. They're both capital M's. It's dominant. We use the capital M. All right, if we've got homozygous recessive, which is what the yellow is it going to be uppercase, 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 lowercase, lowercase, lowercase? Show me with your fingers. Point. Which one's it going to be? Yeah. All right. So I see people doing lowercase, lowercase. Again, it's recessive. So I use the lowercase. And they're going to be the same. So they're both lowercase and lowercase. We'll set up and do the Punnett square tomorrow. Right? We do tomorrow. And we'll talk about the genotypes and the phenotypes. All right.